Electrophysiology, Wikipedia audio. Electrophysiology, F. S. Physis, nature, origin, and A. Logia is the study of the electrical properties of biological cells and tissues. It involves measurements of voltage changes or electric current or manipulations on a wide variety of scales from single ion channel proteins to whole organs like the heart. In neuroscience, it includes measurements of the electrical activity of neurons, and, in particular, action potential activity. Recordings of large-scale electric signals from the nervous system, such as electroencephalography, may also be referred to as electrophysiological recordings. They are useful for electrodiagnosis and monitoring. Electrophysiology is the science and branch of physiology that pertains to the flow of ions in biological tissues and, in particular, to the electrical recording techniques that enable the measurement of this flow. Classical electrophysiology techniques involve placing electrodes into various preparations of biological tissue. The principal types of electrodes are: the principal preparations include definition and scope. If an electrode is small enough in diameter, then the electrophysiologist may choose to insert the tip into a single cell. Such a configuration allows direct observation and recording of the intracellular electrical activity of a single cell. However, this invasive setup reduces the life of the cell and causes a leak of substances across the cell membrane. Intracellular activity may also be observed using a specially formed glass pipette containing an electrolyte. In this technique, the microscopic pipette tip is pressed against the cell membrane, to which it tightly adheres by an interaction between glass and lipids of the cell membrane. The electrolyte within the pipette may be brought into fluid continuity with the cytoplasm by delivering a pulse of negative pressure to the pipette in order to rupture the small patch of membrane encircled by the pipette rim. Alternatively, ionic continuity may be established by perforating the patch by allowing exogenous pore-forming agent within the electrolyte to insert themselves into the membrane patch. Finally, the patch may be left intact. The electrophysiologist may choose not to insert the tip into a single cell. Instead, the electrode tip may be left in continuity with the extracellular space. If the tip is small enough, such a configuration may allow indirect observation and recording of action potentials from a single cell, and is termed single-unit recording. Depending on the preparation and precise placement, an extracellular configuration may pick up the activity of several nearby cells simultaneously, and this is termed multi-unit recording. It allows for integration of microfluidics, which enables automatic compound application for ion channel screening. The system is accessible for optical or scanning probe techniques. Perfusion of the intracellular side can be performed. As electrode size increases, the resolving power decreases. Larger electrodes are sensitive only to the net activity of many cells, termed local field potentials. Still larger electrodes, such as uninsulated needles and surface electrodes used by clinical and surgical neurophysiologists, are sensitive only to certain types of synchronous activity within populations of cells numbering in the millions. Other classical electrophysiological techniques include single-channel recording and amperometry. Electrophysiological recording in general is sometimes called electrography, with the record thus produced being an electrogram. However, the word electrography has other senses, and the specific types of electrophysiological recording are usually called by specific names, constructed on the pattern of electro++ graphy. Relatedly, the word electrogram often carries the specific meaning of intracardiac electrogram, which is like an electrocardiogram but with some invasive leads rather than only non-invasive leads. Electrophysiological recording for clinical diagnostic purposes is included within the category of electrodiagnostic testing. The various XG modes are as follows. Optical electrophysiological techniques were created by scientists and engineers to overcome one of the main limitations of classical techniques. Classical techniques allow observation of electrical activity at approximately a single point within a volume of tissue. Essentially, classical techniques singularize a distributed phenomenon. Interest in the spatial distribution of bioelectric activity prompted development of molecules capable of emitting light in response to their electrical or chemical environment. Examples are voltage-sensitive dyes and fluorescing proteins. After introducing one or more such compounds into tissue via perfusion, injection, or gene expression, the one- or two-dimensional distribution of electrical activity may be observed and recorded. The consumable biorecognition elements, the electronic readout device with embedded artificial intelligence. Intracellular recording involves measuring voltage and/or current across the membrane of a cell. To make an intracellular recording, the tip of a fine microelectrode must be inserted inside the cell, so that the membrane potential can be measured. Typically, the resting membrane potential of a healthy cell will be -60 to -80 MV, and during an action potential the membrane potential might reach +40 MV. In 1963, Alan Lloyd Hodgkin and Andrew Fielding Huxley won the Nobel Prize in Physiology or Medicine for their contribution to understanding the mechanisms underlying the generation of action potentials in neurons. Their experiments involved intracellular recordings from the giant axon of Atlantic squid, and were among the first applications of the voltage clamp technique. Today, most microelectrodes used for intracellular recording are glass micropipettes, with a tip diameter of 1 micrometer, and a resistance of several megohms. The micropipettes are filled with a solution that has a similar ionic composition to the intracellular fluid of the cell. A chloride silver wire inserted into the pipette connects the electrolyte electrically to the amplifier and signal processing circuit. The voltage measured by the electrode is compared to the voltage of a reference electrode, usually a silver chloride-coated silver wire in contact with the extracellular fluid around the cell. In general, the smaller the electrode tip, the higher its electrical resistance, so an electrode is a compromise between size and resistance. The voltage clamp technique allows an experimenter to clamp the cell potential at a chosen value. This makes it possible to measure how much ionic current crosses a cell's membrane at any given voltage. This is important because many of the ion channels in the membrane of a neuron are voltage-gated ion channels, which open only when the membrane voltage is within a certain range. Voltage clamp measurements of current are made possible by the near-simultaneous digital subtraction of transient capacitive currents that pass as the recording electrode and cell membrane are charged to alter the cell's potential. Classical electrophysiological techniques. The current clamp technique records the membrane potential by injecting current into a cell through the recording electrode. Unlike in the voltage clamp mode, where the membrane potential is held at a level determined by the experimenter, in current clamp mode the membrane potential is free to vary, and the amplifier records whatever voltage the cell generates on its own or as a result of stimulation. This technique is used to study how a cell responds when electric current enters a cell, This is important for instance for understanding how neurons respond to neurotransmitters that act by opening membrane ion channels. Most current clamp amplifiers provide little or no amplification of the voltage changes recorded from the cell. The amplifier is actually an electrometer, sometimes referred to as a unity gain amplifier. Its main purpose is to reduce the electrical load on the small signals produced by cells so that they can be accurately recorded by low-impedance electronics. The amplifier increases the current behind the signal while decreasing the resistance over which that current passes. Consider this example based on Ohm's law. A voltage of 10 mV is generated by passing 10 nanoamperes of current across 1 mO of resistance. The electrometer changes this high-impedance signal to a low-impedance signal by using a voltage follower circuit. A voltage follower reads the voltage on the input. It then instructs a parallel circuit that has a large current source behind it and adjusts the resistance of that parallel circuit to give the same output voltage, but across a lower resistance. This technique was developed by Erwin Neyer and Bert Sakmann, who received the Nobel Prize in 1991. Conventional intracellular recording involves impaling a cell with a fine electrode. Patch clamp recording takes a different approach. A patch clamp microelectrode is a micropipette with a relatively large tip diameter. The microelectrode is placed next to a cell, and gentle suction is applied through the microelectrode to draw a piece of the cell membrane into the microelectrode tip. The glass tip forms a high-resistance seal with the cell membrane. This configuration is the cell-attached mode, and it can be used for studying the activity of the ion channels that are present in the patch of membrane. If more suction is now applied, the small patch of membrane in the electrode tip can be displaced, leaving the electrode sealed to the rest of the cell. This whole cell mode allows very stable intracellular recording. A disadvantage is that the intracellular fluid of the cell mixes with the solution inside the recording electrode, and so some important components of the intracellular fluid can be diluted. A variant of this technique, the perforated patch technique, tries to minimize these problems. Instead of applying suction to displace the membrane patch from the electrode tip, it is also possible to make small holes on the patch with pore-forming agents so that large molecules such as proteins can stay inside the cell and ions can pass through the holes freely. Also, the patch of membrane can be pulled away from the rest of the cell. This approach enables the membrane properties of the patch to be analyzed pharmacologically. In situations where one wants to record the potential inside the cell membrane with minimal effect on the ionic constitution of the intracellular fluid, a sharp electrode can be used. These micropipettes are again like those for patch clamp pulled from glass capillaries, but the pore is much smaller so that there is very little ion exchange between the intracellular fluid and the electrolyte in the pipette. The resistance of the micropipette electrode is tens or hundreds of mO. Often the tip of the electrode is filled with various kinds of dyes like lucifer yellow to fill the cells recorded from, for later confirmation of their morphology under a microscope. The dyes are injected by applying a positive or negative, DC or pulsed voltage to the electrodes depending on the polarity of the dye. An electrode introduced into the brain of a living animal will detect electrical activity that is generated by the neurons adjacent to the electrode tip. If the electrode is a microelectrode, with a tip size of about 1 micrometer, the electrode will usually detect the activity of at most one neuron. Recording in this way is in general called single-unit recording. The action potentials recorded are very much like the action potentials that are recorded intracellularly, but the signals are very much smaller. Most recordings of the activity of single neurons in anesthetized and conscious animals are made in this way. Recordings of single neurons in living animals have provided important insights into how the brain processes information. For example, David Hubel and Torsten Wiesel recorded the activity of single neurons in the primary visual cortex of the anesthetized cat, and showed how single neurons in this area respond to very specific features of a visual stimulus. Hubel and Wiesel were awarded the Nobel Prize in Physiology or Medicine in 1981. If the electrode tip is slightly larger, then the electrode might record the activity generated by several neurons. This type of recording is often called multi-unit recording, and is often used in conscious animals to record changes in the activity in a discrete brain area during normal activity. Recordings from one or more such electrodes that are closely spaced can be used to identify the number of cells around it as well as which of the spikes come from which cell. This process is called spike sorting and is suitable in areas where there are identified types of cells with well-defined spike characteristics. If the electrode tip is bigger still, in general the activity of individual neurons cannot be distinguished, but the electrode will still be able to record a field potential generated by the activity of many cells. Extracellular field potentials are local current sinks or sources that are generated by the collective activity of many cells. Usually, a field potential is generated by the simultaneous activation of many neurons by synaptic transmission. The diagram to the right shows hippocampal synaptic field potentials. At the right, the lower trace shows a negative wave that corresponds to a current sink caused by positive charges entering cells through postsynaptic glutamate receptors, while the upper trace shows a positive wave that is generated by the current that leaves the cell to complete the circuit. For more information, see local field potential. Principle and mechanisms. Electrographic modalities by body part. Amperometry uses a carbon electrode to record changes in the chemical composition of the oxidized components of a biological solution. Oxidation and reduction is accomplished by changing the voltage at the active surface of the recording electrode in a process known as scanning. Because certain brain chemicals lose or gain electrons at characteristic voltages, individual species can be identified. Amperometry has been used for studying exocytosis in the nervous and endocrine systems. Many monoamine neurotransmitters, e.g., norepinephrine, dopamine, and serotonin are oxidizable. The method can also be used with cells that do not secrete oxidizable neurotransmitters by loading them with 5-HT or dopamine. Optical electrophysiological techniques. Intracellular recording. Voltage clamp. Current clamp. The patch clamp technique. Planar patch clamp is a novel method developed for high-throughput electrophysiology. Instead of positioning a pipette on an adherent cell, cell suspension is pipetted on a chip containing a microstructured aperture. A single cell is then positioned on the hole by suction and a tight connection is formed. The planar geometry offers a variety of advantages compared to the classical experiment. With this electrophysiological approach, proteoliposomes, membrane vesicles, or membrane fragments containing the channel or transporter of interest are adsorbed to a lipid monolayer painted over a functionalized electrode. This electrode consists of a glass support, a chromium layer, a gold layer, and an octadesilmer-captane monolayer. Because the painted membrane is supported by the electrode, it is called a solid supported membrane. It is important to note that mechanical perturbations, which usually destroy a biological lipid membrane, do not influence the lifetime of an SSM. The capacitive electrode is so mechanically stable that solutions may be rapidly exchanged at its surface. This property allows the application of rapid substrate/ligand concentration jumps to investigate the electrogenic activity of the protein of interest, measured via capacitive coupling between the vesicles and the electrode. Sharp electrode technique. The bioelectric recognition assay is a novel method for determination of various chemical and biological molecules by measuring changes in the membrane potential of cells immobilized in a gel matrix. Apart from the increased stability of the electrode-cell interface, immobilization preserves the viability and physiological functions of the cells. BERA is used primarily in biosensor applications in order to assay analytes that can interact with the immobilized cells by changing the cell membrane potential. In this way, when a positive sample is added to the sensor, a characteristic, signature-like change in electrical potential occurs. BERA is the core technology behind the recently launched Pan-European FoodScan project about pesticide and food risk assessment in Europe. BERA has been used for the detection of human viruses, veterinary disease agents, and plant viruses in a specific, rapid, reproducible, and cost-efficient fashion. The method has also been used for the detection of environmental toxins, such as pesticides and mycotoxins in food, and 2,4,6-trichloroanisole in cork and wine, as well as the determination of very low concentrations of the superoxide anion in clinical samples. A BERA sensor has two parts. A recent advance is the development of a technique called molecular identification through membrane engineering. This technique allows for building cells with defined specificity for virtually any molecule of interest, by embedding thousands of artificial receptors into the cell membrane. While not strictly constituting an experimental measurement, methods have been developed to examine the conductive properties of proteins and biomembranes in silico. These are mainly molecular dynamics simulations in which a model system like a lipid bilayer is subjected to an externally applied voltage. Studies using these setups have been able to study dynamical phenomena like electroporation of membranes and ion translocation by channels. The benefit of such methods is the high level of detail of the active conduction mechanism, given by the inherently high resolution and data density that atomistic simulation affords. There are significant drawbacks, given by the uncertainty of the legitimacy of the model and the computational cost of modeling systems that are large enough and over sufficient timescales to be considered reproducing the macroscopic properties of the systems themselves. While atomistic simulations may access timescales close to, or into the microsecond domain, this is still several orders of magnitude lower than even the resolution of experimental methods such as patch clamping. Minimum information standards or reporting guidelines specify the minimum amount of metadata and data required to meet a specific aim or aims in a clinical study. The minimum information about a neuroscience investigation family of reporting guideline documents aims to provide a consistent set of guidelines in order to report an electrophysiology experiment. In practice, a mini-module comprises a checklist of information that should be provided when a data set is described for publication. Extracellular recording, single unit recording, multi-unit recording, field potentials, amperometry, planar patch clamp. Other methods: solid supported membrane-based, bioelectric recognition assay, computational electrophysiology, clinical reporting guidelines.